So first, just want to thank Harmon and everyone for letting me present this work. And the title of my talk is Building Accurate Sequence-to-Affinity Models from In Vitro Binding Data Using Feature Reduce, which is this is our follow-up method to matrix reduce. So my first 4 slides are motivation slides, and I want to motivate how accurate affinity modeling of protein DNA interactions can help to facilitate current systems biology approaches to model gene regulation and also to model gene regulatory diseases like cancer. And as I'm sure a lot of people in this room know, that different cancers respond differently to different chemotherapy treatments. Now, as Steve talked about yesterday and other people have mentioned, so Todd et al. In 1999, they were among one of the first groups to show that genome-wide expression profiling of gene expression can be used to correctly classify different cancer phenotypes. For example, here, the 2 phenotypes are acute lymphoblastic leukemia and acute myeloid leukemia. And they showed how just doing, looking at the average expression profiles of just a handful of genes can be used to correctly classify with a high degree of accuracy different cancer phenotypes. Right. Now, a question that we can ask, of course, is what is driving the differences in gene expression between these 2 different cancer types? And to answer that question, we're going to look at the central dogma of gene regulation. And the central dogma, of course, is that DNA makes RNA, makes protein. Of course, the central dogma was proposed before microRNAs and all these non-coding RNAs entered the picture and kind of muddied this up a bit, right? So first, if we look at the first layer in this process, which is transcription, we have in this process a very complicated regulatory network of molecular machinery, right? Where you have acetylases and deacetylases, methyltransferases, et cetera, that modify the chromatin and open up DNA-encoded genes on the genome so that then transcription factors can then bind to near the TSS, can recruit more proteins that then start the initiation complex, right? Open up the double-stranded DNA, create the initiation bubble, and then you have the transcription initiates, and then you have maintenance of the elongation bubble as the transcript is being created, right? The pre-mRNA transcript is being created. And after a transcript is being created, first let's talk about the regulators. So the regulators of this process include, you have the DNA binding transcription factors, you have chromatin remodelers, and so on. Now, in the translation layer, after the pre-mRNA has been created, we have another complex network of molecular machinery, especially in eukaryotes, right? Where you have the addition of the poly A tail, the addition of the 5' cap. You also have the spliceosome machinery, which recognizes splice sites on the pre-mRNA, then cuts out, splices out the introns, and then creates your mature messenger RNA, which then can leave the nucleus. And in the cytosol, there we have additional molecular machinery, such as microRNAs and RNA-binding proteins, that then regulate the ribosomal machinery that then translates the mature messenger RNA in order to create protein. So this is a sort of story we've heard a few times now. And the regulators of this process include the RNA-binding proteins, microRNAs, and so on. And what my research, most of the research in our lab has been focusing on are the DNA binding transcription factors and the RNA binding proteins that regulate both the transcription and the translation of genes. Okay? And let's first focus on transcription factors. Now, in transcription factors belong to sometimes very large families, okay? And typically, these transcription factors that belong to a particular family bind to very similar sequences on the DNA. And Markle and Quake showed in 2007 that FO4 and CBF1, which are two BZIP proteins, BHLH proteins, bind to very similar sequences. Basically, different variants of the E-box motif. The E-box motif has got this has this central CAC GTG hexamer core, right? And then you have these variable flanks on the two sides. And it's just the variation on the flanks that differentiates the specificity. So FO4 prefers some flanks, while CBF1 prefers other flanks. And what they showed in their paper is that basically that the genomes have evolved to capitalize on these small differences in specificity. And FO4 actually regulates a very different set of genes than CBF1. And that these genes are also in very different pathways. So the take-home message here is that small differences small quantitative differences in specificity lead to very large qualitative differences in gene regulation. So let me say that again. That small differences in specificity lead to very big differences in gene regulation. And so hopefully this helps to motivate why, if we want to truly understand the underlying molecular mechanisms of transcription factors binding to their binding sites near the promoters or their downstream target genes, we need to understand these small differences in specificity between transcription factors and the same TF families that bind to very similar sequences. Okay, so that's the goal, right? The goal is to basically create databases similar to like JASPAR and TransFac, right? Where for all the transcription factors in the human genome, we have 2,000 plus transcription factors that maybe 2,500 that we can understand their specificities, and we can, looking at genomic sequences, understand where the transcription factors are binding to the DNA and why, and how transcription factors can differentiate when they can bind and when they shouldn't bind, and understand sort of the complex circuitry of transcription. In order to do that, a bunch of groups have invented a bunch of in vitro binding assays. Oh, I want to mention too, if anybody has any questions at any point, please raise your hand. Okay? Yes?
This sort of departs from the previous slide, kind of departs from the assumption that the binding motif is, these small variations are responsible for the big differences, right? But there are things like the glucocorticoid receptor, which has at least an identical motif to the mineralocorticoid receptor, but they have widely different functions.
Yes, yes.
And it has to do more with which cells can hydrolyze a given ligand to the receptor and which cells can't. So it's sort of another level of control where this assumption is violated, right?
No, it's absolutely. There's other means of regulation. One is, of course, is ligand binding in cofactors. So, which I'm going to talk about later. I'm going to talk about cofactor complexes, which is really, I think, the story is getting much more clear that in higher metazoans, including us, that much of the regulation is determined by cofactor complexes. These proteins aren't binding to the DNA alone, right, to regulate downstream targets. They are binding with a complex, recruiting other proteins. So they're really sort of, transcription factors really chaperones to bring down a whole set of proteins that come down and sit on the DNA and modify the chromatin, open up, right, the DNA so that then all this transcriptional machinery can come in and start the initiation problem. So, so in order to sort of build up this database of the specificities of different transcription factors, there's been a lot of work done by different groups. I'm going to talk about basically all these groups, all these different methods fall into two main categories. The first sort of generation of in vitro binding assays, which was pioneered in the Bullock lab at Harvard, was built on a microarray technology. And in this method, so you have these microarrays, and they're different from your typical expression arrays in that the spots, the probes, are double-stranded DNA as opposed to single-stranded DNA. So you have double-stranded DNA probes, and in the you basically just throw on your protein of interest, right, and have a binding reaction. The proteins, often it's actually just the DNA binding domain, but sometimes it's full length. But in any way, they have an alpha GST tag, right, fusion at the end, and this alpha GST tag fusion at the end of the protein is recognized by a specific antibody, and the antibody has attached to it a fluorophore. So after letting the binding reaction reach equilibrium, you throw in the antibody, and then you, with the use of a fluorescence scanner, you then take a picture, a snapshot of the microarray, and what you have here is for every single spot here represents how much fluorescence intensity do you have, which is hopefully, if you're in the low free protein concentration limit, Harman talked about a couple of days ago, then this level of fluorescence intensity, right, is proportional to level of occupancy of how much protein is bound to the DNA at that spot. Right? And if you're in the low free protein concentration regime, the level of occupancy at the spot is proportional to the affinity that the proteins have for the probe sequence at that spot. Okay? And I want to talk about their different PBM designs. Okay? One of the most common designs is it's called the UPBM array, which stands for Universal Protein Binding Microarray. And it's called universal because these DNA sequences are specifically designed so that they have a 25 base pair primer region, which is near the surface of the slide, and that primer is used to double strand the single-stranded DNA oligos after the single-stranded DNA oligos are grown on the array, and the primer is used to double strand it. But then at the edge, at the free ends of these probes, the last 35 base pairs have what they call the variable region. And these variable regions have been designed, specifically designed, so that across the entire microarray, which has about 44,000 spots on it, right? Across the entire microarray, every temer is represented exactly once. So if you were to look at temer windows and you slide them, you look at this temer and then slide one base pair and look at the next temer, and then slide one base pair and look at the next temer. If you were to do that across all the variable regions for all 44,000 probe sequences, and you were to count, you would, at the very end, you would see that every temer was counted exactly once. So the goal for these UPBM arrays is to characterize the specificity of any protein across binding sites that are at most 10 base pairs long. Okay? Now, of course, there's a limitation, right? Because what if your protein binds to more than 10 base pairs, and then you've got a problem. And that's where the sort of the new generation of in vitro binding assays are much more applicable. Any questions about PBM? Okay, good. So this is sort of the first generation, and so the next generation of binding assays is, of course, is built on high-throughput sequencing, right? And HT-Selects and Select-Seq, these are two very similar methods. I'll be talking about Select-Seq. later in the talk, and in the R lab we're going to do later on this morning, we'll be analyzing CLEC-seq data and characterizing the binding specificities of two EXT Hox protein complexes. So here, it's a little bit different. Now, what you have is you have these very large random libraries. Again, you have the DNA oligos on the two sides of the DNA oligos are going to be, in our case, Illumina adapter sequences. And then in the middle, you have the variable region, right? And you want your library to be large enough so that you have good coverage of all possible, chemers of a certain length, just like the PBM design. And so now the reactions occur just in solution, and our double-stranded DNA oligos are not attached to any slide or any spots. And then after the binding reaction, we don't need any fluorophores or antibodies. Now we just sequence. Oh, we select, of course, for the protein of interest. And then the DNA oligos that we pull down with the protein, we then throw into a sequencer. And so the data that we get from this sort of the new generation of binding assays is for sequence reads. Let me FASTQ files. So actually, there's the very different data that's analyzed to calculate these binding specificities. The first generation, of course, we use roughly normally distributed, real valued fluorescence intensities, right? And in the new generation, we have these Poisson distributed integer valued read counts, right, what people have talked about in the last 3 or 4 days. And because of that, there's different strategies that we use to analyze this data. Okay? Any questions? Yes?
So in any case, you need to wait the antibody to cool down the protein.
There are different methods to do this. The method we'll focus on is the CLEC-seq method that we're going to use in the lab. We use an EMSA-gel shift. So the reason why the EMSA-gel shift is advantageous is because we're actually looking at protein complexes, right? And so we were interested in the EXT and an EXT Hox dimeric complex. So with using an EMSA-gel shift, we can cut out the band that corresponds to that heterodimeric complex. And then we can elute that out, and so that we can basically filter out any of the monomer binding or homodimer binding. So that's an advantage. Okay. And that's one of the major advantages also this new method has over the PBM design. If you were to throw in 2 proteins, right, that are cofactors onto the PBM design, you get all this binding, but the binding would be confounded. Some of the binding would be, from monomeric binding from protein 1 or protein 2. Some of the binding would be, homodimeric binding of, protein 1, homodimerizing, so on. But if all you're interested in is you want to know what's the binding specificity for the heterodimeric complex, it's very difficult, right, to build, because all your binding data is confounded from all these other protein complexes. Okay. Any other questions? Yes?
So how do you know the in vitro binding reflecting the in vivo binding side?
That's a good question. No, that's a good question. Basically because, actually I'm going to show some of that data. People basically do experiments, and they basically want to say, okay, how well does, so say I do an in vitro binding assay, and I get I calculate these binding specificities for a protein. And I want to know, well, how well do these binding specificities reflect, right, the in vivo environment, the binding in the in vivo environment? Typically what you do is you say, okay, how well can this motif differentiate the sequences corresponding to my ChIP-seq peaks relative to random genomic DNA, right? And one of the things that people have talked about, and also in previous lectures, is a lot of people also try and build affinity models from in vivo binding data, like ChIP-seq data, right? And it's really, it's understandable why, because there's so much ChIP-seq data, right? And so certainly, if people have ChIP-seq data and they want to know from that, you know, okay, so what's the sequence the protein is binding to? There are additional challenges because the in vivo environment is so messy. All right, you have the major artifact you have to deal with is the issue of DNA accessibility, right? That what you're looking at in the in vivo setting through ChIP-seq data is highly biased because not all the genomic DNA is equally accessible. You have these hotspot regions that people have talked about, right? And it's difficult to normalize that artifact out. So that's why using in vitro binding data to build your specificity models is so much easier, because in the in vitro setting, you don't have to worry about chromatin state, right? Or DNA accessibility. And you just, all the DNA is equally accessible, and so it's much easier than to say, okay, you know, if I have, you know, more higher occupancy here than I have higher occupancy here, it's solely because of differences in affinity to the underlying DNA sequence, right? I don't have to worry about, well, in the in vivo setting, it's a combination of, well, how accessible first was that DNA? And then after that, you say, okay, then it's dependent upon the underlying DNA sequence. Yeah.
So the reagents that you guys begin with here are like DNA libraries and then protein in solution in another bottle, and then mix them, or?
Yeah. So this is in solution. Of course, you have much more DNA library than protein, right? You want to be in a low free protein concentration regime. There's no saturation of the higher affinity binding sites.
And you do it, is it one protein at a time, or protein mixtures, or?
Well, it depends on what you're looking for. If you know, you basically have it's usually 6. Well, we would do in 1 to 10 picomoles of protein, right? If it's just looking at want to look at monomeric binding or homodimeric binding, then you just put in the one protein. But if you want to look at complexes, then you put in different, you know, concentrations. And you know, you brought up a good question. What is sort of a little bit of an art, what we've done is, say you want to look at the you're interested in a heterodimeric complex. Well, many times, say you have protein 1 and protein 2, and you want to look at the heterodimeric complex. Many times, protein 1 and protein 2 will also homodimerize, right, with itself. And they will do this at different rates. Like protein 1 may be more apt to homodimerize than protein 2. And so you basically, you play with the different concentrations to get the complex that you're interested in assaying. So if you're interested in the heterodimeric complex, right? If protein 1 homodimerizes very readily, right? And protein 2 does not. What you do is you put in a lot more protein 2 than protein 1, so that way you're pushing the reaction into the heterodimeric complex, because that's the complex you care about. That's the most binding you want to get. So that way, when you do the Empson gel shift, that's where most of your DNA, right, has shifted towards, so that you'll have good library size to sequence. That was a very technical thing, but I hope that made sense. Yeah.
I wanted to ask, this binding to, so this dimerization, does it occur prior to binding to the DNA, or because sometimes you could imagine that you need the binding to the dimerization, right?
Yes. You're absolutely right. Sometimes proteins will readily heterodimerize or homodimerize, right, in solution. Other proteins actually require the DNA in order to heterodimerize or homodimerize. And in that scenario, the theory is that the DNA sort of serves as a scaffold, right? And it helps induce the complex, the formation. And so what you have there is sort of the DNA, serves as as a scaffold to make that complex occur. Examples for P53, right? That DNA helps to create the tetrameric complex. Okay, so now that we've sort of characterized the binding data and classified them in two major groups, let's now talk about the models, right? The affinity models. And some of this is going to be reviewed because Harman's already gone over a lot of this material. So first of all, we're going to talk about the positional independence model that we use in the Bussemacher lab. And there actually are multiple positional independence models out there, and Harman's talked about them. But the one that we use in Harman's lab is this position-specific affinity matrix, or PSAM. And as he's talked about, this is a positional independence model that assumes that every position within the binding site, right, contributes independently and additively to the overall binding free energy, right, that the protein has for a particular DNA sequence. And these matrices, which are different from PWMs, and these matrices, they are parameterized in affinity space. So they're parameterized in affinity space, and in order to calculate what is the affinity that a protein has particular, this is a 10 base pair binding site, so for any 10 base pair binding site, you multiply the marginal affinities for each column. So you can see that for the highest affinity sequence, and this is, by the way, this is the affinity motif for MIBL2, which is a member of the MIB family of transcription factors. In order to calculate the relative affinity for a sequence, here the highest affinity sequence, it's simply just 1 times 1 times 1 times 1 times 1 times 1. So the highest affinity sequence has a relative affinity of 1, right? And as you look at single point mutations away from this highest affinity sequence, you can see basically that there is a multiplicative hit in affinity. So here we mutated the G to a C, and so now we had about a hit of about, now the new sequence has roughly one third the affinity that the original highest affinity sequence had. So now it's just 1 times 1 times 1 times 1 times 0.29 times 1 times 1 times 1 which equals 0.29. Okay? And this positional independence model sort of forms the backbone of a lot of the work that I'm going to show next, in which we build on this positional independence assumption to look at higher order features and see if there are higher order sequence features, right, that on top of this positional independence model that helps to increase the accuracy of the affinity model. So let's talk about that next. Oh, so before we do that, actually, I have a pictorial representation of how the new feature-reduced method builds affinity models from PBM data. So first I'm going to show you how we build affinity models from PBM data, and then I'm going to show you how we build affinity models using CSHL data. Remember from the second slide that, well, I guess the sixth slide, that the binding data from PBM is very different from the binding data we get from CSHL. Right? So here's my cartoon representation of one spot on a PBM array. Okay? And there's 44,000 of these spots. This is one spot. And what I'm showing here, my happy faces and sad faces, these are the proteins. Okay, this is the protein that's binding to these binding sites, which are characterized in red. The dark blue sequence, these are variable regions, variable DNA regions, and this light blue, these are the constant regions which contain the primer sequence. And the analogy that I like to use in characterizing this binding is the bead on a carpet. Okay? And in this cartoon here, the carpet is sideways. The carpet is sideways, and the carpet's going, this is the surface of the carpet, and the carpet's going out this way. So these are the free ends of the probes, and these are the ends that are attached to the surface. And the artifact that, one of the major artifacts in PBM data that we had to model in order to greatly increase the accuracy of our models is the fact that the proteins, which are like the beads, right, they have difficulty in getting into the closely compacted carpet of double-stranded DNA oligos for any particular spot. So that's why these proteins are happy, is it's easy for them to, they don't have to go very deep into the carpet, right? It's easy for them to access their binding sites. This protein is sad because its binding site is way deep into the carpet, right? And it's very difficult for it to reach in here and bind to this binding site. So, in a more technical explanation, right, what's really happening in the PBM environment, the binding reaction, is that there's this closely compacted, close compaction of these double-stranded DNA oligos in each spot is creating local free protein concentrations, so that as you go deeper into the carpet, the free protein concentration is a gradient. It's becoming less and less. Okay? And so, actually here, I'm going to go through this, but here is actually a depiction of the amount of occupancy, the amount of binding that you see across all the probes for this PBM data. So you can see what's happening here is where's position 0, right, is the position free end of the probe, right? In position 34, you're attached to the primer. And you see this, first of all, you see there's this, on the negative strand, there's a huge amount of what we call overhang binding, okay? And that's actually depicted here. This cartoon here is showing overhang binding, where the full binding site actually isn't even represented on the probe sequence, right? It's actually missing part of the binding site over here. But because there's a double-stranded break here, you know, that changes the biochemistry, right? And so, because of that, sometimes the proteins may like, you know, they may like having part of the binding site, but then the double-stranded break changes the chemistry, and it can still bind there. So that's what's being depicted here. And then as you go deeper into the carpet, you see less and less and less binding for the exact same binding site. So if you take the exact same binding site and you start here, you know, you get a lot of binding, and as you move the exact same binding site deeper into the carpet, you get less and less occupancy. Right? And this is one of the main artifacts of PBM data that we had to model. So quickly, let me just show you, just go through pictorially how the feature-reduced method builds these affinity models. So first, we perform a robust multivariate regression over all 8-mers to get the highest affinity seed sequence. So here's the highest affinity seed sequence. And then from that, we then also perform multivariate regression to then look at, you know, all single point mutations away from that highest affinity seed, and we can construct this positional independence model, this PSAM. Right? And now if we look at the, I'll push the wrong button. If we look at the self R squared using our positional independence model, we see that that our biophysical model, just using the positional independence model, we explain 64% of the variance in the data using the model, which is pretty good. That's pretty good. But now the next step is, now with this affinity model, we can now perform regression and then find out, okay, using this affinity model, now let's look at 10 base pair windows across all the probes, and we can build this occupancy bias profile, right? And this is where we can see for the same binding motifs, there's a huge difference, right, in how much fluorescence intensity we see on this for each spot. And then what we do is, now given this occupancy bias profile, right, we can now go back, use this, and refit our affinity parameters. So we use this to increase the accuracy of our affinity model. And then we say, okay, now we have a more accurate model, then we can go back and refit the occupancy parameters, occupancy bias parameters. So we then do this iteratively until we get convergence, right? And the model, the parameters don't change. And at the end, what we see is the final R squared using our occupancy bias profiles now is increased. Now we explain 78% of the variance in the binding data. So this is the pictorial representation of basically how feature reduce builds affinity models from PBM.
How much did how much does that figure change when you iterate?
Oh, you're talking about this?
No, the one on the left. Do you have like a little higher problem? Those, as you iterate, the values for each position change slightly, right?
Yeah.
And so how much change do you see at the beginning versus the end of that?
It depends. It totally depends on, you know, how extreme the bias is. The bias is very extreme. This can change a lot. Okay. And actually, in this example, it changed a lot because if you don't take in this bias effect, you can see that the binding that occurs, all the binding that occurs here at the free end, okay, and it doesn't really. You can have lots of point mutations in the highest affinity site. It doesn't even matter. It's only the binding that occurs at the free end that's dominating, right, your affinity parameters. So if this occupancy bias profile for a particular protein is more uniform, then you see very little change in the affinity parameters. And there's one thing, we'll take a question. One thing I want to say is that for each protein, what we see is these occupancy bias profiles change. Okay, they change considerably. And what we notice is that we think the relationship is really very dependent upon how big is the protein, right? What's the molecular weight? And how big is it? I mean, if it's a big bead, right, it's hard to get into the carpet, and it's a really small bead. So these occupancy bias profiles change considerably depending on the protein. Yeah.
Is there kind of similar to the pair idea in the sequencing, like array with which free promoter adapters for the other side pairing two arrays, which you can using two arrays to reduce the artifact available?
You know, people do that. So there's a lot of the labs, they actually will build two different PBM arrays, right? And then they'll compare. And actually, the Dream 5 competition, which I'm going to talk about in a couple more slides, the first part of that competition actually had to deal with two different PBM arrays. You train on one, and you affinity model on one of the PBM arrays, and you predict the probe intensities on the other array. So that's one way to try and see, well, you know, the affinity model that you build, you know, how much is that characterizing actually protein DNA specificity, and how much of that is actually capturing some sort of PBM artifact. So I'll talk about that in actually in 2 slides. Okay. Any other questions? All right. So now that we've done this sort of pictorial representation, we're going to be a little more rigorous now and say some equations, right? And the nice thing is that this first slide, Harmon's actually gone through all of these, right? So this is just a review. So it's the biophysical model is, of course, we have protein, right, and the sequence, and then you have binding to create the protein sequence complex, right, protein DNA complex. And you have a K-on rate and a K-off rate. And then the binding association constant for the protein has for the sequence, which is obviously is also 1 over the KD, which is the dissociation constant, right? It's just the rate of K-on over K-off. It's that ratio. And then Harmon showed how, right, this is also can be characterized using this Boltzmann equation, right, where delta G is the binding free energy, the free energy of Gibbs free energy of binding, in terms of RT, right? R is the gas constant and T is absolute temperature. And in this simple reaction, right, all of this, the binding association constant can be characterized as basically what's the concentration of the protein DNA complex over, right, the product of concentration of the protein free, the free protein, and the concentration of the unbound DNA sequence. Okay? So this is fine and dandy. However, the data that we get, right, the data that we actually have, we get the ability to affinity models from, right, is occupancy data, okay, which is the level of binding, and also what's the probability, right, that the protein is bound to the sequence. And that probability is characterized as, now instead, the denominator has a sum, right? We have it's the concentration of the complex over, right, the two possible scenarios, either the binding site is bound or unbound, right? And that's your level of occupancy. And now we can see is, by plugging in Ka sub s, we can see that actually we get this sort of nonlinear function in which occupancy can be calculated based solely on the free protein concentration and the binding affinity that the protein has for the sequence. That's how we get this nonlinear function. Okay? And what this nonlinear function does is it basically models the saturation effect. Right? As you increase the free protein concentration, right, what happens is you get saturation, where you end up saturating the higher affinity sites. And the occupancies become, right, more similar between a high affinity site and a medium affinity site. And so a lot of times we say is that what happens is that you get this sort of squenching, right, where now your level of relative occupancy is not proportional to the level to your relative affinities. Okay? So Harmer's talked about this a lot. One analogy, one I like to use, is this might be helpful, is let's assume that you have a protein, right? Protein A, and performing a binding reaction with a bunch of random DNA oligos, right? And you're using a particular concentration, say concentration level. And now what happens is that, let's say you keep increasing the concentration, right? Until now, in your library, all the highest affinity sequences, right, are bound by the protein. You've saturated the highest affinity sequence, right? Now what happens when you add on you increase the concentration even more of the protein. And now you increase protein concentration even more. Well, what happens? Well, all your highest affinity sequences are already bound by the protein. So protein, you don't see any more increase in occupancy of the highest affinity sequence. So what does the protein do? Well, it can't go to the highest affinity sequence. So now the new protein that you add, as you increase the concentration, starts binding to the next highest affinity sequence, right? Or next highest affinity sequences. So you can see what's happening is now your level of occupancy is not proportional to your level of affinity.
Just want to make sure that there's no confusion, because this is not really how it works, that the highest affinity sites get occupied first, right, and then the lower one. They all get bound. It's just that, a little bit more when they're higher affinity and lower affinity. Right, so this sounds a bit like the electron shell model where you fill up the lower shells.
Oh, sorry. Did I say that? I did something to
It's
say.
an analogy, but it's not really what
Yeah.
happens.
Let me restate that. You're adding on more protein and protein, right? In the beginning, of course, what happens is all the occupancies are increase, right, in proportion to the affinities. The affinities the proteins has for each one of those sequences. That's what happens in the beginning, until all the highest affinity sequences are bound. Okay, then what you see is after all the highest affinity sequences are bound, now as you add on more protein, now you get the saturation effect. Okay, where now you can increase occupancy at the highest affinity sequence because they're already bound, but you increase occupancy at all the other sequences, and that's now no longer is your relative occupancy proportional to relative affinity. Sorry, thank you for correcting that. Yes?
Maybe you can explain a bit. The way you're describing makes me wonder. I understand how you model this in the in vitro situation, they're equally represented in the library that goes into making the autologous pool. But in vivo, the situation is very different because not all hexamers are represented equally, and not all hexamers are accessible equally, right? So do you later on model that situation in vivo, or is that just too complicated? Because
We do try and model that in vivo. And you're right. That's one of the reasons why building these affinity models from in vivo data is so difficult. Because your in vivo genome is not random DNA. Genomic DNA has huge sequence biases. GC content, for example. You have GC islands, right? CPG islands in front of genes, and on top of that, for a lot of genomes, so the human genome, you have these what do they call? It's called GC. If you look at a particular chromosome, you'll see regions where it's GC rich, and these regions are very large and encompass many genes. Isocores, thank you. I was going to say isoforms, but that's not right. Isocores. You have these GC isocores. So across any genome, you have these GC isocores where you'll have a genomic sequence which is very GC rich, and it'll comprise of many, many genes, and then AT rich, and it'll comprise many, many genes, and then GC rich, and these are called GC isocores. So not only do across the entire, if you take the entire genome, do you have nucleotide bias, but even locally on chromosomes, there's difference in GC content, and even on partitions of chromosomes, you have big differences in GC content. So, okay. Hopefully now we corrected my scenario. So make sure that everyone understands that in my story, right, as you increase the protein in the very beginning, from you have no protein, you're increasing the protein concentration, the relative occupancies are proportional to relative affinities, right? Then once you saturate the highest affinity binding site, right, the moment after that happens and you add more protein, now you get this saturation effect. All right, now let's talk about, now we're ready to look at how feature reduce builds on this biophysical model. And what I start off with, we're looking at affinity space, and I've written this formula to correspond to PBM data, right? The data that we get these are fluorescence intensities of a probe sequence. Fluorescence intensity of a probe sequence. And what we assume is that the fluorescence intensity of any probe sequence is proportional to, in a linear relationship, the level of occupancy on that spot, that probe sequence, for the protein, level of occupancy of that protein. And alpha corresponds to the gain, right? The gain in fluorescence intensity based on occupancy. So alpha, basically is how intense, right, is the scanner. And beta, this is our baseline intensities when there's absolutely no binding. Okay, so start off with a simple formula. And now what we do is, we do two things. First is that we do this sliding window technique that people have talked about in the past, where these DNA, the DNA probes are much longer, right, than the binding sites. So what we assume is that the overall occupancy for a probe is equal to the sum of every binding site on that probe. So we basically look at, say if our binding site is 10 base pairs long, we look at the first binding site, 10 base pairs, and we slide one position, we'll look then at that binding site, and we just sum over every sliding 10 base pair window, we sum over the level of occupancy for each one of those, and that gives us the total occupancy for the entire probe. So that's what the summation is doing. And now what I've done here is basically just replaced our occupancy variable now in terms of the function in terms of the free protein concentration and the binding association constant from the previous slide. And then lastly, the next step is basically I've taken the association constant and I've split it into two terms. The two terms are the sequence-specific binding association constant, right, and the non-specific binding association constant. Because every protein actually has some affinity for DNA sequence. Because DNA is, overall is slightly negatively charged. And so all proteins, transcription factors, have some slight, completely sequence non-specific binding affinity for DNA. Okay? And this is the formula that we use in feature reduce in order to, by physical underlying formula we use to then analyze both PBM data and CLX data to build these affinity models. Any questions?
Alpha set by the experiment? By the gain?
We fit alpha. We fit all of these actually. Okay. So now let's talk about the inference schemes. Here, what I've done is I've taken the previous formula and I've now added into, we fit for the parameters, looking at this maximum likelihood least squares, right, formulation. So to solve for alpha and beta and the sequence-specific binding association constant and non-specific binding association constant, we want to minimize, find the parameters that minimize this function, right, the least squares, between our measured effluence intensities and our formula for the occupancy for every probe. And there's different ways to solve this. The saturation model is where we assume that we're in the low free protein concentration regime, right? So we assume that that the association constant is much less, right, than 1, much less than the KD. And in there, we solve basically the linearized version of this equation. And the non-saturation model, here is where we don't make the assumption that there's no saturation. And this is where we actually solve this full version, the nonlinear version. And actually, I reversed that. This is where we solve the nonlinear version. This is where the non-saturation model is where we look at the linearized form of this equation, where we assume that the free protein concentration is much less than the KD. So, and there are different ways to solve the linearized form of the equation. You have ordinary least squares, non-negative least squares, and the method that's worked the best for us, and this is sort of a recurring theme, right, that you've seen this week, is that the robust regression method works the best. The reason why is because the PBM data is very noisy. It has lots of outliers. So by using the robust regression method to solve for our affinity parameters, we get the most accurate models looking at cross-validation across PBM experiments. And then finally, I'm talking about at the end, the Poisson regression model is what we use in order to model CLX-seq read counts. So the CLX-seq data, again, we have Poisson distributed integer value reads, and so it makes sense to use the Poisson regression. Any questions?
So you don't see the biological variation in this interesting
Oh. Yeah, the problem, we're interested in that. The thing is that we don't have replicate data. We don't have replicate CLX-seq data. So, as of now, it's difficult to solve, right, for the extra biological variance. We don't have replicate data. It's probably there, we can't solve for it, so.
So maybe just to add to that, there's also, with RNA-seq, you're trying to make a call about differential expression for every individual gene, right? So you need to make sure you're not calling false positive p-values. Here we're just trying to infer some parameters that act across all the probes on the array, so we don't have to worry so much about modeling the errors in individual genes as accurately as possible, because we're not trying to compute the p-value for every individual gene. So we're a little more less sensitive to this issue.
Yeah, no, that's true. Any other questions? Okay. So, first of all, one of the things that we noticed was that when we compared our affinity models built on PBM data, and we compared that to third-party KD estimates, one of the things that we noticed right away is that for those PBM experiments in which we saw sort of this strong bias to bind the free end of the probes, right? And as binding sites went closer to the surface, we saw reduced binding. We noticed that when you had this sort of negative slope here, that typically our affinity models had really good match to KD estimates from third-party assays like SPR, right, or Matomi. And then here for FO4, you see this is totally reversed. We actually have reduced binding at the free end of the probes and more binding close to the surface of the slide. And we noticed there was a correlation whenever we saw this, that our affinity estimates had a reduced, right, match to third-party KD measurements. So we believe what's happening here is that basically whenever we see this, that these are examples of failed PBM experiments, and that what we think is happening is there's a wash step in between after you perform the binding reaction, right, and before you take a picture using the scanner of the fluorescence intensities. That was probably what's happened here, is that there was too much of a wash, right, and that we washed away a lot of the sequence-specific binding on the free end of the probes. And so because of that, we have in this experimental data, we probably had a huge reduction, right, in sequence-specific binding. And then what happens then, what's happened is now your signal-to-noise ratio is much less favorable. And so because the reduction in signal-to-noise ratio, that's why our affinity estimates have a worse, have a huge decrease in accuracy. So this is an interesting finding. Now we're ready to, now that we've gone over sort of the positional independence model, we're going to look at higher order sequence features. So let's go back to the original PSAM. This is the same PSAM that I showed before for MIBL2. And there again is the highest affinity sequence, right? And here is our point mutation that we made before, right? That G to a C at position 1 in the binding site, right? Now, what happens though that if we look at the observed affinity was 0.44, right? And what if for every time that we saw across the sequence, whenever we saw that there was a CC, right, in the center of the binding site, what if we saw that there was the same sort of, CC in the center, what if we saw that there was the same sort of discrepancy where we were off by roughly about 50% between our estimated affinity and our observed affinity? There's where we can say, okay, what's happening there is that there's a dependency, right, in the matrix. And that here, our positional independence assumption is breaking down. It doesn't hold. So in order to correct the positional independence model, what we do is we add a dependency correction. We say for any time you have a CC, right, in the center of the binding site here, position is -1 + 1, we have a dependency correction where we increase the affinity estimate by 50%. And so people call this is a dinucleotide dependency correction. And if we do this now, if we perform this sort of calculation, and we look at all possible dinucleotide dependencies between any two positions, right, we can actually construct a whole, array of dinucleotide correction terms. And that's what the next slide is. So now what I've done is, here we've basically looked at here we're looking at actually just adjacent dinucleotide dependency terms. Look at all the adjacent dinucleotide dependencies and say, how much of a correction is there? And then if we take that set and then we sort them so that the biggest dependencies are at the top, and then as you go down the list, the dependency correction becomes less. We get a plot like this. So what this shows actually is, first, we put the PSAM up top here. This is the positional independence model, right? And now, here are the adjacent dynamic dependency corrections, and they're sorted based on so they have decreasing dependency correction as you go down the list. If we look at the top, then dependency corrections, we see that actually it's in the center of the binding site that the positional independence assumption breaks down the most, right? And for example, we see that if you have the CG here, you actually get an increase in affinity, a 50% increase in affinity relative to what the positional independence model estimate gave you. Okay? Any questions? One of the things too is any dinucleotide dependency correction can either increase your affinity estimate or decrease it. So a green row here, right? A green row signifies that this, whenever you see this dinucleotides together in the binding site, a green row corresponds an increase in affinity relative to the positional independence model. A red row corresponds to a decrease in affinity. So your correction is actually a decrease. This make sense? So now, let me talk about when we throw on all these adjacent dinucleotide dependency corrections, how well does that increase the accuracy of our affinity model? And then to do that, I'm going to show you the results from a recent comparison study that was done by Matt Rorrock in Tim Hughes' lab. And in this comparison study, they took 36 basically all known published methods, right, to analyze PBM data and to build protein DNA specificity models. And there were two parts of this comparison study. The first part is with the in vitro cost validation performance study, where there were two PBM designs, okay, and the difference between the designs are the probe sequences are different, okay? And you would train on one PBM design, build your affinity model, and then using this affinity model, you then predict the probe intensity of the second design, which was held out. You didn't know the intensities of the second design. All you had were the probe sequences, okay? And then the second part of this performance comparison study is where you would take the affinity model that you built here in the first part, take the affinity model, and then using totally independent ChIP-seq data, right, you would measure how well does your affinity model, how well can it discriminate the underlying DNA sequences that correspond to the ChIP-seq peaks? How can it differentiate those sequences from random genomic DNA? Okay? And so let me just show you the results. So here are the results from the in vitro performance comparison. And you can see there actually, this competition consisted of 66 different experiments for 33 different proteins. Because each protein, you did the experiment twice, one on one PBM array design and one on the other. And we were happy to see that actually that our new method, the feature-reduced method, actually was performed the best. Okay. And now, if you look at the in vivo performance comparison, what we did is for a handful of those 33 proteins in which there was either good ChIP-seq or ChIP-exo data, we did. They calculated these UROC scores. Has anyone presented what a UROC score is yet? How many people know what a UROC score is? Okay, yes. So this is the area under a receiver operator curve. Okay, and what these are non-parametric measurements of how well can you differentiate one set of sequences over another set of sequences. And here what we want to measure is how well can this method, right, properly label sequences that should be bound by the protein, and these are sequences that correspond to ChIP-seq peaks, right? How well can your method properly label that those sequences should have higher affinity than random genomic sequences. Okay? And basically, what you want is a UROC score of 1. So if you have a UROC score of 1, that means you have a perfect classifier, right? And you are 100% accuracy in labeling that this is a ChIP-seq sequence, right? The sequence corresponds to ChIP-seq peak, and it always has higher affinity. You label all those that have higher affinity than all the random genomic sequences. And then typically these scores go down to 0.5. If you have a UROC score of 0.5, that means that your classifier does no better than random guessing. Okay? There's no better random guessing. So, again, that we saw, we were happy to see that we came in first. And one of the things that's interesting is that in this study, actually, we do not see at all a significant difference in the mean UROC score between the independence model and our dot-nucleotide dependencies model. There are different theories as to why that is. One is what is the sensitivity, right, of this comparison? How many ChIP-seq peaks do they look at? But I'm going to show you in the next couple of slides the analysis we do to show that our dinucleotide models, are better than our positional independence models for a lot of proteins. And here are these reference rows here. I just want you to know these were put in sort of as a theoretical upper bound, right, of how well you could do. These are methods that are building specificity models from the ChIP-seq data, right? So these two methods, they have a huge boost because their test data is the same as their training data, right? And so that's why, that's why their scores are so high. But the nice thing is actually, the feature-reduced models that are built on the PBM data, right? They're trained on the PBM data. When you create the UROC scores on test data, they get very similar scores to these reference methods. Yes.
So would I interpret for like ZFX, the global score from each chip means that something biological is happening that is preventing it from finding its desired sequence?
You mean right here?
Yeah.
Yeah. I'm not sure what's happening there. My guess is it probably picked the wrong seed. The thing is that the meme method, right? It gives you many motifs, right? And it has to pick one of those many motifs, and because it's a stochastic method, right? That what it means is you run it multiple times. If you roll them multiple times, you don't get the same motif every time, right? So sometimes it'll give you the wrong motif, and you have to run it many times, and even then it's difficult to say, right, which one is the correct motif and which one is the second motif, the third motif, which could have a very similar score given by meme. There's a challenge there picking which one is the right binding motif. Okay, so this is just showing that our method works well. Okay? And here I just want to show, one of the things that we're very interested in is, again, I think one of the reasons why people are investing so much time and effort, right, in these in vitro binding assays and groups like us who are building these in vitro binding affinity models, is that we want to use this data and use these in vitro binding affinity models to understand what's happening in the cell, right, in the in vivo environment. And so this is a measurement to show, using this, our affinity model we built using PBM data for CBF1, if we compare that to in vivo occupancy for 955 E-box motifs in the yeast genome, we get a, I think, pretty good match. Right? And again, this is the in vivo setting, which is a lot messier than the in vitro setting. But we get a root mean squared error of 0.075, which is pretty good. Okay. So now we're going to switch over to select-seq. Take a break?
Maybe it's a good time to take a short break, maybe do some questions, but then
Okay.
it's kind of a different, right?
Okay. Yes.
So can you go back to the previous slide? So you're basically using the in vitro to predict whether the in vivo gives you the same result, or you're comparing the in vivo result to the in vitro result to see the match.
Yeah. So.
Is this a prediction, or is this just a?
Yeah. So on the x-axis, this is what our affinity estimates are, given our affinity model built on PBM data. So x-axis these are our relative affinity estimates built on PBM data.
For each specific sequence.
For each specific, in each one of these points corresponds to a 10 base pair E-box motif, where they all have the same CAC GTG hexamer core, and all that's varying are the two positions on the left and two positions on the right, the flanking sequence. And then on the y-axis here, this is the measured relative occupancy enrichment. For you did two ChIP-seq experiments. One was control, and then one was, after doing pull down for for CBF1 binding. And so this is a relative enrichment in the ChIP-seq reads. Okay. And so for each one of these 10 base pair binding sites, we're looking at it's the mean. What was the mean ChIP enrichment across? Because the yeast genome has multiple copies of each one of these E-box motifs. And one thing I want to explain too, see how there's a curve here? The curvature is, we actually fit what is the in vivo free protein concentration. So by fitting the in vivo free protein concentration, we actually can see that there's a curve here which is illustrating that there is some saturation here. We do have some saturation of binding for the highest affinity E-box motifs. And then the red lines correspond, this is our error, right? In an ideal world, all of our points would be on the green line, green curve. And, but we're not perfect. Yeah, either one.
I had a question about the dependency model.
Okay.
Why do you think that the dependencies are always higher in the center compared to the?
Why are all the dependencies?
Like when CC in the center of the sequence, is that higher compared to when it's in the periphery, or is that?
I'm sorry, I didn't hear the last part. Higher than what?
So the correction that you do.
Yeah.
Like the next random decreasing dependency correction.
Yeah.
Do you see a certain pattern of the correction always being higher if the sequence dependent, but like it's position dependent if it's in the center of the sequence versus on the periphery?
No, we don't. We don't see that. For this protein, that's the case, but for other proteins, it's not the case. Matter of fact, for CBF1, it's the opposite. For CBF1 and FO4 and MAX and a lot of the HLH proteins, all the binding specificities in the flanks, right? And they all bind to the same CAC GTG, core hexamer in the center. And so we see all the dependencies are in the flanking positions. So it's not the case. This distribution of where the positional, where does the position dependence assumption break down, is not the same. I think it varies across different transcription factor families.